This is the video manual for liquid-liquid extraction for Group E. Liquid-liquid extraction, or LLE, is a chemical separation process that utilizes differences in the chemical potential of a solute in two different liquid phases to cause mass transfer of the solute from one phase to the other. The purpose of the experiment performed in this lab was to separate propionic acid from a light oil solution using water as a solvent. Continuous LLE typically has two inlet streams and two outlet streams. One inlet is the feed solution containing the solute to be removed, and the other inlet is the solvent used to remove the solute. The outlet stream containing the original solution phase is called the raffinate, and the outlet stream containing the solvent phase is called the extract. Frequently, one of the two phases is aqueous, and the other is organic. In this experiment, the feed solution and raffinate are organic, and the solvent and extract are aqueous. In a packed column, one liquid phase is the continuous phase, and one is the dispersed phase. During operation, the continuous phase fills most of the volume of the packed column, while the dispersed phase flows through it in droplet form. Literature sources and experimental results suggested that an aqueous continuous phase produces a better separation than an organic continuous phase, so the aqueous phase was used for experimental trials in this lab. The pilot apparatus shown here is the pilot equipment that we used in the lab, and it contains organic and aqueous feed tanks used to store the two liquids before being pumped through the column by the organic and aqueous feed pumps. These pumps are calibrated to pump 0 to 250 milliliters per minute of aqueous feed or organic feed each. The transfer of the propionic acid from the organic phase to the aqueous phase takes place in a glass column of diameter of 0.05 meters that is filled with 1.2 meters of cylindrical glass packing. The aqueous phase is fed to the top of the column, and the organic phase is fed to the bottom of the column. The less dense organic phase forms droplets that float up through the continuous phase in counter-current flow. As the two phases flow through the column, the raffinate is collected in the organic return tank, and the extract is collected in the aqueous return tank. Before beginning the column operation, each of the valves on the apparatus must be checked to ensure that they are in the correct positions and will not expel liquid from the apparatus in undesired locations. When operating with water as the continuous phase, first fill the column with water up to the interface line by turning on the water pump on the apparatus touchpad. After filling the column with water up to the desired interface line, shut off the water pump and turn on the oil pump until the rest of the column is filled with oil. To run a trial, the aqueous and organic pumps must be turned on via the touchpad. The flow rate of each stream can be controlled individually to collect data on different operating conditions. During each trial, the interface between the aqueous and organic phases is maintained at a constant level by continually adjusting the interface valve located near the top of the column. In the video shown, you can see my hand adjusting the valve to maintain the interface at the desired location. To collect data during trials, open the UOP5MK2 software installed on the computer next to the column. After launching the software, click the Start/Stop button circled to begin recording the data. The most pertinent data recorded in the program is the measurement of the propionic acid concentration in the aqueous extract. To observe the data being recorded during trials, click on the Graph button and check the concentration out box to the right. Those two buttons are circled in the picture below. The graph can be used to indicate when the system has reached steady state. Once the system has reached steady state, stop the data collection using the Start/Stop button and export the data to Excel using the Save As button. Once all trials have been completed for the day, close the software and begin the cleanup procedure. For the cleanup procedure, press the water pump button that shows on the touchpad screen to send most of the organic phase to drain into the organic return tank. The next step in the procedure is to drain the LLE column through the bottom valve into a pitcher. The valve is showing in the picture on the left with an arrow. Use a separatory funnel then to separate the aqueous phase, organic phase, and the inner phase from each other. Place the aqueous phase into the aqueous return tank to be titrated later, the inner phase into the organic waste tank, and the organic phase into the organic return tank in the pilot equipment. The acidic aqueous solution in the aqueous return tank in the pilot equipment must be titrated as part of the cleanup procedure as well. In this lab, 1 molar of sodium hydroxide should be used to titrate the aqueous return. The first step of this process is to take a sample from the aqueous return tank from the valve shown in the right picture with an arrow, and titrate it to calculate the required amount of base that must be added to the aqueous return tank to neutralize the solution. Before titrating, add a color indicator to the tank to make sure that the titration is successful. Then, pour the neutralized aqueous return down the drain in the lab through the valve at the bottom of the tank while flushing the drain with water from the eye wash station. Finally, the control panel must be turned off when all the previous procedures are done by pressing the stop button and turning the main power breaker to the off position. Here are some instructions on titration. Titration is used in this experiment to determine the propionic acid concentration in a sample. This involves the slow addition of 0.1 molar of sodium hydroxide to a sample of a known volume but unknown acid concentration. Color indicators added which turns pink in basic solutions and can be used to identify the point at which the solution is neutralized. 2 types of samples must be titrated in this experiment. The first type is to calibrate the concentration measurements recorded by the UOP5 MK2 software, which are based on a system of water and acetic acid. In order to calibrate the concentration measurements to a system with propionic acid, aqueous samples must be collected at the bottom of the column at different steady-state extract concentrations. In our lab, these samples were titrated, and the concentration determined by titration was plotted versus the concentration on the display to determine the factor at which the concentration on the display were inaccurate. The second type of titration that needs to be performed is to determine the organic feed acid concentration. For each new batch or feed, a sample must be collected from the organic feed tank to allow the aqueous color indicator to disperse throughout the sample. Add a small amount of surfactant to the oil in order to do that, then perform the titration as normal. But pay special attention to the milky white sample, as the indicator turning pink is more difficult to see. This concludes our video introduction of LLE. Thank you for watching.